you to our EICT Academy's Global Online Faculty Development Program on Advanced Optimization Technique using MATLAB.
Thank you, Dr. Nanda. I'm audible? Hello?
Yes, you are perfectly audible. So participants, let me introduce you to Dr. Sitanshu Sekhar Sahu. Dr. Sahu has received the PhD degree from NIT Rourkela. He completed his postdoctoral research from Oklahoma State University, USA. He is presently working as an associate professor in the Department of Electronics and Communication Engineering at Birla Institute of Technology, Mesra, Ranchi. He has been a recipient of Department of Foreign Affairs and International Trade, DFAIT, Graduate Fellowship from Canada Government, with which he worked at University of Western Ontario. His research interests are biomedical signal processing, bioinformatics, machine learning, and computer vision. So, Dr. Sahu, welcome. You can share your screen.
Okay. Thank you, Dr. Nanda, for such a nice introduction. Let me share my slides. Just confirm.
Dr. Sahu, presently we have nearly 170 participants in this platform. And for other participants also, this will be live streamed at YouTube.
All right. Okay. Is it visible?
Yes. Please make it full screen. Dr. Sahu, all participants will be in the chat mode.
Okay.
They will not have unmuting and direct interaction, that will not be there. But if you ask them over chat, they will reply on the chat. And they have any questions, they will put it in the chat and you can read the chat and answer them.
Okay, so
that will be at the end also in your lecture. Okay.
Okay, perfect. So should I start?
Yes, please.
All right. Okay. So today I'll be discussing on the topic ANOVA PSO based feature selection method for Parkinson disease detection. So basically I'll be discussing on PSO, particle swarm optimization algorithm. So how it will be used and the ANOVA context to extract or select the features, meaningful features for the prediction of Parkinson disease. So my talk will be the flow will be like this. So I'll be discussing a little bit about what is the Parkinson disease, what is the challenges, how we process detection of Parkinson disease, and there itself again I'll discuss in the optimization framework how the Parkinson disease process can be looked upon as a optimization framework. Then particle swarm optimization, how it is being used to solve the disease detection. So this will be the flow of my talk. So let us start with the Parkinson disease. What is Parkinson disease? So basically, Parkinson disease is a neurological degenerative disorder which happens in the central nervous system, which is caused because of the loss of neurotransmitter called dopamine, which is deposited across the substantia nigra portion in our midbrain. If you look at the left side figure, basically in the This one is the cross section of a human brain in the middle region. So this is called your substantia nigra. In the substantia nigra, so here actually the dopamine neurotransmitters they secrete, and it is deposited across here. If you see this figure in the top, so here you can see the deposition of the neurotransmitter dopamine across here. So now if it is start losing depositing over here, the dopamine cells, so then it creates the disease called Parkinson disease. So Parkinson disease basically a motor related disease. So basically what is the symptoms we observe when it happens, the Parkinson disease? So the symptoms are tremor. One of the most prevalent symptoms is the tremor. That means the shaking of hand and shaking of your head, so continuously. So you don't have a control over the motor activity of your hands. Okay. Then postural instability. So you cannot stand still in a perfect way. That related to the instability of the postural. Then the bradykinesia. So we cannot walk in a proper manner because of the malfunctioning of the neuromuscular things inside the body. Then rigidity. So will be you cannot walk properly. So you cannot move your hands or gates basically. These are the rigidity happens. So you cannot stand. You cannot walk properly. Similarly, the loss of smell also happens. Dysarthria of voice also is getting deteriorated. So dementia, so memory loss kind of things. Then anxiety, these are some of the symptoms. But the confirmatory symptoms are the tremor, postural instability, bradykinesia, rigidity. These are the things, and these are the confirmatory states. Like once someone have gone into Parkinson disease, so these are the symptoms happens. Now once someone is having gotten the Parkinson, so there is no cure till now. So there is no drug available in the market which can stop or which can undo the effect of Parkinsonism. Only thing that the availability of the drugs like the Syndopa, Levodopa, so that can delay the severity condition a little bit ahead, but you cannot cure the Parkinson disease. There is no as a drug available for that. Only thing that we can go for a management of the Parkinson disease so that it can be delayed. The severity condition can be delayed much better, so that we can live in a normal lifestyle in the further of our life. So now what is the diagnosis? What are the diagnoses till now available? So the most confirmatory diagnosis is the MRI scans or PET scans of the human brain, where they looks the deposition of the dopamine receptors on the mid central brain part of the brain. So this then again is the gait impairment. So the postural instability, the bradykinesia, that can be also examined by the doctors. They ask just to walk for a while or stand, some kind of exercise they prescribe, and through that they try to guess whether you have a Parkinsonism or not. And then the other one is the diagnosis is the speech impairment. As you said, speech also, your voice is also getting deteriorated. So there is a speech dysphonia or dysarthria happens. That is the indication of also Parkinsonism. Similarly, there is a sleep disorders also, olfactory probes also, smell disorder also, another test generally the physicians they perform. But the most confirmatory tests are the PET scans or MRI scans, which gives an indication, confirmation indication that you have a Parkinson. But MRI scan, PET scan is a very costly affair, and expertise also has to be there, which is not available everywhere in the rural area, including the rural area also. And not all the parts of country there is no availability of this kind of facility. So again, so that also takes a lot of time, and a lot of money is required to go for a confirmatory state of Parkinson in the imaging technology. So then people start off like, what could be the other modality from which we can detect the Parkinsonism in early stage? So one of the modalities is the speech. As I said, the speech also getting varied, or it is getting deteriorated or changed before the happening of the Parkinsonism also, right? So if you could able to detect or grasp the pattern of change of the voice, basically we can guess an idea so that whether it is a Parkinson voice or is a healthy voice. So that's the point of research. Nowadays a lot of research is going on from the speech modality, how to diagnose a Parkinson's. Also there are some other modalities also available, you have a handwritten pattern also, handwritten patterns, and your RBD patterns also people have used. But mostly nowadays people are going for the speech modality, because why? The speech is very easy to acquire and easy to process. It's a non-evasive also, and is an indication, the early indication of Parkinson's. So it has been seen that 70 to 90% of the patients with Parkinson's disease, they have a difficulty in articulation and phonation, which is indication that, okay, if you could able to get the pattern or observe the phenomenon of change of voice over the time, then you could get an guess that whether it is towards affecting towards the Parkinsonism or not. So that's the basis of detection of Parkinsonism through the voice. Now, if you see how the speech is related to the motor activity, and how the Parkinsonism affect the speech. Now, if you look at the middle figure in the brain, so the cross section of brain, the midbrain region, which is actually controls the motor activity inside the body. Basically, see, it is triggered at some different parts of a human brain, so which controls the motor activity of different body parts. Now you see the leg portion over here, and the hand person, and the mouth, the tongue. So these are the portion of the central mid part of our human brain, so which actually controls the movement of these organs. Now when something goes wrong in this region, basically the substantia nigra region, if something goes wrong here, and the generation of the signal which actually triggers the activity of the motor organs inside our body, so that start deteriorating, that starts malfunctioning. So what they has to do in a proper way, so that could not be functioned. So that signal is now getting deteriorated. Once the Parkinson happens, that signal starts deteriorating, and since it could not receive in a proper way the motor organs of our speech production system, so then our speech also getting deteriorated. Now if you see the production of the speech in the right side of the figure, you could see the diaphragm, which is the source of energy, like the air is coming out of the lung, then it is passes to the esophagus. In the esophagus, if you see here, there is a tiny kind of a skin here, so it is called your vocal fold, so that get vibrated. So once it is vibrated, so certain amount of vibration, so that certain amount of frequency is being generated over here, and that frequency and that air of certain frequency is passed through the mouth cavity, and that mouth cavity, which is constitute of the lip, jaw, jaw, teeth, everything, so that modulate that passes of the air, that modulate the air, and it gets some kind of harmonics is being produced over here, and then the air is coming out of the mouth, what we observe as a speech or voice of signal. So again, so here, once in the substantia nigra part, once the motor dysfunction starts because of the loss of generation of the dopamine, so it start giving a weak signal or malfunctioning signal in the motor activity regions. And since that signal could not be reached in a proper way to this the motor organs of our mouth cavity, so then our voice also is getting deteriorated accordingly. So now once the voice is deteriorated, so if you could able to catch that deterioration or that change in the voice, so that could be give us a idea whether it is of a Parkinson voice or a healthy voice. So that's the idea basically. How do you go for detection of Parkinson disease from a speech modality? Now, if I go on modeling this, a speech, an electronic model, I could see here the muscle force. The air is coming from the lungs, and then it passes to the esophagus. So, which is can be thought of as a tube. Okay, and somewhere in between the tube there is a tiny structure over here which is called your vocal cords. Now, this vocal cord is start vibrating. So basic fundamental frequencies of any particular sound is getting generated over here, and then it passes through the pharynx cavity and the mouth cavity, where it is getting modulated by several motor organs, just like your tongue, teeth, lips. So these are some motor organs, and that motor organs start modulating the passage of the air, and that sound is getting coming across the mouth cavity. So you can think of this as two different tube-like structure. One is called your vocal cord tube, another is your mouth cavity tube, okay, or the vocal tract tube. So this could be thought of as a model of two different systems, digital systems. So you can see here uh, the air coming from the lungs can be thought of as a periodic impulse trains, okay, which is getting modulated across the system of vocal fold or the glottis. Okay, so then the output of this glottis can be fed to the vocal tract, which can be act as a filter. Again, it is modulated across this filter, and this vocal tract output is coming as a speech signal. What you observe, the voice or the sounds, different sounds. So the speech signal can be thought of as a the convolution of the impulse trains with the vocal fold, then with the vocal tract filters. So this is an electronic model how we can model the speech signal. So then from the speech signal itself, can we identify different patterns? What are the different patterns present in the speech signal? So that which getting deteriorates basically, which getting deteriorates by the progression of the Parkinson disease. So that deterioration, if you could able to catch, then we can say whether it's a Parkinson voice or it's a healthy voice. Now see what affects the Parkinson. What is the effect of the Parkinson disease on the vocal fold and vocal tract? What is a vocal fold? Vocal fold, as I said, it is the vibration of the a tiny structure which is called the vocal fold, which generates the fundamental frequency of any sounds, the fundamental vibration of any different types of sounds. And what is the vocal tract? Vocal tract is the passes, the mouth cavity, where it is getting modulated. So when Parkinson's disease affects, so that is the vocal fold starts improper closing and opening, okay, which leads to a breathiness during the phonation. So that means that is measured by the fundamental frequency, the vibration of that vocal fold. Okay, so that vibration is called the fundamental frequency, which measures the fundamental vibration of the particular different sounds, which is getting deteriorated, like when the Parkinson voice, the Parkinson disease affects. Similarly, the other motor organs which modulates this fundamental frequency, that is the nose, mouth, tongue, and lips, which collectively called the vocal tract, and that vocal tract basically modulates the fundamental frequency, and that generates some Formant frequency, some harmonics of the fundamental. It's called the Formant frequency. So these vocal tract also consist of the movements of the different muscles of mouth, tongue, nose, lips. So since the motor organs start malfunctioning, so the functioning of these motor organs will not be done in a proper way. So that's why there is a change in the Formant frequency of a particular sound also being observed when the Parkinson disease happens. So these two parameters can be simultaneously grasp the changes of these two parameters, then we can say that a particular voice is of healthy or a Parkinson type. So if you look at the spectrogram, this is the raw signals. The upper two graphs are the raw signals of healthy. The left one is the healthy, the right one is the Parkinson of a particular sustained vowel A. So the healthy one, they uttered the vowel A, and the same vowel A also uttered by a person with Parkinsonism. And you see there is a difference in the pattern of the utterance of the vowel A. And the below two are nothing but the spectrogram, how the frequency components of the vowel A changes across the time. We could see here there is a significant difference of the frequency variation of the vowel A of a healthy person and a Parkinson. I could see in the Parkinson, so there is a smearing or smearing of or the spreading of frequency across y-axis. If you see the spreading of the frequency happens, but which is not observed in case of healthy, because healthy person could say the utter the vowel A very pronouncedly. So there is no smearing of the frequency. Similarly, in the time axis, if you could see in the time axis, so the energy is slowly concentrated on a particular region where actually the sound is being uttered. You could see here this region up to 3 seconds of the utterance of the vowel A. So the most of the energy concentrated over here, and then it starts ending. Now you see here in the time domain, so the energy is concentrated in this region only. But if you look at in the Parkinson, the same vowel is being uttered. Now, if you look at the time axis, the energy is again spreaded across the time from 0 to the last time. So this is an indication that, okay, because the Parkinson people cannot pronounce the sound A in a firm way, so that's why the energy is getting also smeared across the time also. Okay, so you cannot stop, you cannot end of the sound in abrupt. Okay, so there is a little bit elongation of the sound energies in case of Parkinsonism, so which is prevalently observed in the time domain also, as well as if you could extract this feature or this deviation, if you could graphs in terms of features, then that features will be used to predict whether it is a of healthy voice or Parkinson voice. So this is of a healthy sound. This is the same sound of Parkinson. Yeah. Now, you see male frequency substral coefficients features. These are the features basically being used very widely in speech signal processing. This feature is nothing but how the different bands of frequencies, different band of frequencies, what are the different energy present. That also gives an indication of the Parkinson voice from the healthy voice. Now, what is a male frequency substral coefficient? A speech signal is there, so we go for a framing of the speech signal, then passes the windowing, and take the first Fourier transform to convert into frequency domain. Then the frequency domain is scaled down with male frequency, certain filter banks. Okay, so band of frequencies, and that the band of frequency you compute the energy. That energy is passed through the DCT cosine transform, and that leads to the coefficients called male frequency substral coefficients. So these MFCC features basically represents the vibrations of the format frequencies. That means which is the harmonics of the fundamental frequency. Okay, so we can capture the vocal tract information, how the modulation of the air happens in the mouth cavity. Okay, so this gives an indication. So MFCC feature is very widely used across all different domains of speech processing. But since it only concentrate on the vocal tract information, but it could not capture the vocal fold information, that is the fundamental frequency. So it means this feature is not sufficient enough to represent or characterize the Parkinson voice. Then with thought of extracting the features in a more meaningful way, by the time frequency domain. So again, the speech signal is framed, then is windowing, then pass through a short time Fourier transform, which is an indication of how the speech energy is varied across the time and the frequency. So that time frequency matrix again, it is a large dimension. So we go for factorizations to reduce the dimension. Okay, so using the non-negative factor. So non-negative matrix factorization. So this NMF gives us two different vectors. One is the base vector, another is the coefficient vector. So this base vector captures the variation across the time, and the coefficient vector captures the variation of the energy across the frequency. So then we extracted some features from the base vector and coefficient vector, just like the frequency movement, the time movement, the sparsity, so how much non-zero components are there, the discontinuity, how much the energy is discontinued across the time, across the frequency, the entropy, and some other statistical features. So these are the features that we have extracted from the base vector as well as coefficient vectors. Then we combine these two feature vectors. We combine to make a spectrotemporal features. So if you see this side, it is the MFCC features, and the right hand side again it is the time frequency based features, and then we concatenate or we combine. That leads to a spatiotemporal spectrotemporal features, which gives a clear cut or advanced feature which represents a much better way the characteristics of the PD speech. Now, when this features being fed into the baseline classifier, machine learning classifier, there is a support vector machine and the random forest. So these are the accuracy in all different speech tags of 5 different vowels: A, E, I, O, and U. It has been seen that the vowel I is giving us highest accuracy of training accuracy, 99% of 95% testing accuracy in SBM, the spectrotemporal features. And it has been the similar pattern. You could see all different vowel tags. Similarly, the A vowel gives the spectrotemporal features, the SBM classifier giving the best accuracy of 88%. Then the vowel E, it gives around 93%, again the spectrotemporal features. Okay, similarly, the vowel O gives us 97% accuracy, and vowel U gives us 91% accuracy. And this one was taken a data set called PC Gita data set, having 50 Parkinson person and 50 healthy persons, and all are Spanish language speakers basically. So this is the accuracy that we have received. But when you test this model with other different speech tags and different languages of same vowel tags, you can start deteriorating. Okay, so it could be you see here the distribution of the samples into two different components using TSNE plot. TSNE component 1 and TSNE component 2, and you see here the red one is the Parkinson, sorry, and the green one is the healthy one. So you see the distribution. Now you see some green dots are mixed with overlapped with the red one, so which indicates that means there's some kind of misclassification could happen, even though if you use any kind of base learning classifiers. It because of the variation of the speech parameter in these features basically, which are getting overlapped. Okay, and moreover, the most challenging task is here. So the less number of samples, only 50 healthy and 50 Parkinson samples are there. and we have extracted the features just like 13 MFCC features and time frequency features around 16. So altogether spectrotemporal features are around 29 features. So each sample have 29 features, and total we have 50 plus 50, 100 samples. So it's quite a difficult task of using such a high dimensional vectors to segregate between two different classes of healthy and PD. That's the real challenge. Okay, so how we can thing is that how we can reduce the number of the dimension of each sample, so that that will give us a good result across the different types of data sets also. That actually leads to a challenging task of optimizing or selecting the most informative feature out of all the 29 features. So that's the point of discussion. So here, so that's why you used ANOVA with particle swarm optimization to extract or the select the meaningful features out of the 29, all 29 features which are more informative, and only keep those extract or the selected features for modeling in the classifier basically. So the objective of the feature selection algorithm is to eliminate the redundant and irrelevant features to maximizing the accuracy of the classifier. Okay, so the objective is to find the best feature subset out of all 29. You don't know like which mixer of all the feature that gives you the best accuracy, you don't know. So it is an optimization problem. So what is the best subset of features out of all 29? What is the best subset of feature that will have a low interclass correlation and high intraclass correlation? Means there should be a clear-cut separation between the classes, and the samples within a class, they should be close enough. Okay, so there should be low intraclass correlation and high intraclass correlation. Okay, so to achieve this, we have used the PSO, the particle swarm optimization, in the ANOVA concept to achieve this feature selection process as an optimization framework. Okay, so what is ANOVA? So ANOVA is analysis of variance. Is basically a statistical test to assess the difference between the means of more than two groups. Okay, so whether the samples of different classes, the mean of these samples are having different or they are similar. So that is indication given by the ANOVA, right? So it compares the mean across the several groups to determine if the observed difference, the difference between the means, is a by chance or this is a genuine distinction happens between them. If they are distinct from each other, if the means are distinct, so that means there is a good for classifications. Okay, so this ANOVA compute measure the F score, the statistical score, which is nothing but the ratio between the interclass variation, the information variation, and the intraclass variation, right? So let us discuss how we compute the F score. The interclass variation basically compute by weighted a square between the classes. Now you see the SBC, which is the interclass variation, is computed by this formula. So here SC represents the number of samples in each class. The C is the superscript, is the class. Okay, so number of samples class C, and MC superscript C, so it represents the mean of the particular class C, and M represents the overall mean of all classes. Okay, so this gives an indication of the variation, the variance difference between the classes, different classes. Okay, and the degree of freedom, the interclass degree of freedom, is nothing but we defined as k minus 1. So k is nothing but the total number of classes. So this squared errors, the squared errors divided by the degree of freedom, interclass degree of freedom, that leads to value called MBC, which is the interclass variation. basically. So then intra-class variation. Let us discuss what is the intra-class variation. In the intra-class variation, so it is nothing but the weighted square errors within the class. Okay. So this is DC. DC is the data samples of a particular class. From each sample, we subtract the mean of that class and that is square of that, and summation of all data and summation of all classes. Okay, so that leads to the squared sum of the within the class variation within class samples. Then the intra-class degree of freedom is nothing but S minus K. So S is the total number of samples and K is the number of classes. So then I take the ratio of WC divided by DWC, that leads to MWC, which is nothing but the intra-class variations. Now the ratio of MBC with ratio of MBC with MWC. So that means the variation of the interclass, the between the class, the variance of the data points between the class divided by the variance within the class. Okay, so that leads to a ratio called F statistics X value, F value. So that F value gives an indication of each feature, whether it could be a significant or insignificant towards discriminating between the classes, right? So now from this F's, the variation of the F values, so we can also discard those features which are having less F value, and those are having higher F value. So we can keep those values of the features. So those are the selected. But this is a greedy selection. Okay, don't know which are having the less F value. That may also contribute towards when it is combined with other features which are having less, which are having greater value. Okay, so that's why we thought of to give some kind of weightage to each of the features, some kind of weightage to each of the features, which gives me a F value which is minimum, Okay, so for that we added a weighted factor over here. You could see here in the inter-class variation, we added here WJ. WJ is the weight factor to each J. J is nothing but the each feature. Okay, so each feature will associated with one weight value. Okay, so this weight value will be used to compute the squared error between the classes. Okay, similarly in the intra-class variation also we use the same weightage values to compute the error. Okay, the square error inside the class points, the intra-class points. Okay, then this F value, which is the ratio of the intra-class divided by the ratio of the inter-class divided by the intra-class variation, take the inverse of that, that as a cost function. So that means I could minimize this cost function in such way that it will give me a minimum variation between the class and maximum variation in intra-class. Okay, minimum variations within the class and maximum variation between the class. So that is my objective, so that all the data samples will be grouped together inside the class, which is separated from different classes. Okay, so that is your that is the optimization framework. Now this optimization framework, so this weights can be optimized, okay, in such way that this CF will be minimum. Okay, this cost function, the F value will be minimum for what value of WJ this CF is will be minimum. So that is nothing but a optimization problem. Now this optimization problem can be solved using any of the optimization algorithms. So here we have used the particle swarm optimization algorithm to find out the optimum value of W that gives me the cost function CF as a minimum. Okay, so cost function is minimum means we have a within class variance less and between class variance higher, so that they are separated from each other. quite good. Then I can use any kind of classifier to classify the samples much better way. Okay, so now what is particle swarm optimization? So it is again, it is optimization techniques first proposed by Ross Everhart and James Kennedy. So one is the engineering professor, another is social scientist. They try to observe what are the behavior of the swarms or the particles or the birds or the fish when they do collective, work in a collective way. So how they optimize their path to the food source. Okay, so that behavior, that swarm behavior is observed by these two scientists, and they have framed a engineering model which is called here a particle swarm optimization. So here the particles are nothing but these are the birds. This could be the bird or the fish, or could be the ants, any kind of social animal. So when they go for foraging or finding out the food particles, so they go for searching in a different direction, while they though maintain a social behavior. So that means they keep on communicating each other. That whoever is having the food source and plentifully available food source, they will give a signal to the others, and others try to follow the path to someone who are having the reach to the maximum food source. So this searching method has been put in engineering framework, what we call the particle swarm optimization. The basic idea over here is each particle is search for the optimum. So that means the food source, where is the maximum food source. So each particle, when they search, they move in a certain direction. Okay, so that's why they have a velocity, they have a certain position, and in what path they are moving, they have certain positions, and each particle remembers the position it was where it had its best result. So that means best food source or the best value of the optimum value of the cost function that we have achieved. So that is called its own personal best. Now again, another behavior of the particles. So when they go for searching of the food, they go in a collective manner. In a group, they go for searching in a group. That is called as a swarm. Okay, and they cooperate each other. So cooperate means they exchange the information about what they have discovered in the places where they have already visited. Okay, so that means they have simple exchanging the information. So they cooperate each other also. So that means a particle has a certain neighborhood associated with it, and each particle knows the fitness of those in its neighborhood. That means who are the other particles, they are in the other swarms are there in the neighborhood, and what is its fitness, what is the best fitness they have. So out of all the neighborhood fitness, which is the best one, that is called the global best. So now this particle try to follow the position of the global best, because they have identified the identity of the food source, or the best, the optimum value of the cost function has been achieved by the G best position. Okay, so that means they will follow the path of the G best. So that these two operations they continuously perform until unless all the particles or all the birds, all the fish, they come together into the where is the plentifully available the food source. So this is the search things, that basically the behavior of the swarm intelligence basically. So it is nothing but you look at a swarm of particles, a group of particles, they fly through the parameter space and search for the optimum. That is the cost function. They search for the cost function. So each particle is characterized by two components. One is a position vector xit, another is the velocity vector vit, because it is flying, it is searching for the food source with a certain direction, which is having a speed and direction, that is a velocity. It is having a velocity vit, and it's a position xit. So each particle has its individual p-based personal base. So he has to move in a certain direction and keep on remembering at what position he got the maximum food source, or the maximum or the optimum value of the cost function. So he remembers that position, that is called your p-based, which is nothing but the best so far he has traveled. each particle has its social knowledge. Social knowledge means, so he has to look at what are the other particles in the neighborhood, and the other particles, what is the p base they have, because they communicate each other, the p base of its neighbors, and the base of all the neighbors is called the global base. So each particle remembers its p base as well as the global base. So based on these two parameters, try to adjust the velocity of the particular swarm or particular particle or particular bird. So it has to update its velocity as well as its position also. This velocity update is like this. So the next velocity vi of t plus 1, the next velocity will be w into vit. So vit is its previous velocity, and w is the inertia weight. So this is a parameter, hyperparameter, which governs the the capability of the search process. So w vit plus c1 into rand is one of the random number generated, into p based at particular particular time instant, minus the position, current time position, okay, from its p based, its own p based, the difference, then plus c2 into rand into g based minus xit. So g based is the global based and xit is the current location. Now p based minus xit, so it indicates its social behavior of its own, and g based minus xit, it's the deviation, which is again another factor of the social behavior in a global sense. Okay, so using these parameters, each particle try to update its velocity vit plus 1. Okay, so here the hyperparameters are the w, the inertia weight, and the social constants are c1 and c2, okay, which governs the social activity of the p based, its own based so far, and the global based of including all the neighborhood. Okay, so now the particle, it sense that, okay, so there is a place where the best cost function or best food source is available, is try to update its velocity using this formulation, then try to change the position, and the position will be nothing but xit plus 1 equal to xit, the old position, plus the updated velocity vit plus 1. Okay, so try to update the position like this. We got the mean path to travel to the global best by looking at calculating this vit plus 1, and then adding to the current position. So we'll try to move in this direction, this line, the pink line, and we'll move the direction to reach to the global best, because the global best position, we got the best food source. So this is the phenomenon that every particles they follow to change its position and its velocity, and using this updated velocity to change the positions to the direction where there is a availability of the food source or the optimum of the cost functions. This is the algorithm, and this algorithm is getting repeated and repeated until unless all this after all this the birds or the swarms, they collectively move to a one place where is the plenty of food source. That means they have found the optimum value. Okay, so if you look at this concept of ANOVA in in the feature selection process, so here is the flow chart. So I've extracted the spectrotemporal features, the combination of the MFCC with time frequency features. That means total 29 features are there. So we initialize the parameters of the PSO, that means the number of particles, the social constants C1 and C2, then W max and W minimum, the inertial weights of the velocity vector, the maximum iteration. Then we initialize the population, initialize the position of each particle xi, then initialize the velocity of each particle vi, then calculate the fitness function of each particle, then find out its p based and the global based, and then look at then update the inertial weights according the iteration of according to the iteration, update the inertial weights, then use this inertial weight along with the social constants U1 and C to update the velocity and position, then again compute the fitness, right, fitness of the new position, then check whether the new position is better than the old one. Okay, if it is yes, then update the p based. Similarly, calculate which is the best among all the neighborhoods, that will be replaced by the g based, then increment the iterations. Then check whether we have reached to the global best, okay, which is my optimum goal, or the maximum iteration has been reached. Okay, until then it go on updating iteratively the velocity and the position until unless they reach to a common place where is the global optimum is there. Okay, once it is there, reach the global point, then it will be stopped, and then compute the threshold, that is the mean of the inertia, mean of the weightage given to all the features. Okay, then apply this threshold which is above the mean threshold, then we choose those are your important features, and then those important features, the selected features, will be used in the SVM and random forest or any other baseline classifier to classify between healthy and Parkinson disease. So this is the optimum framework using the ANOVA concept to choose or select the best feature which gives us variations of information between the classes, as well as maintaining a minimum variation within the class samples, right? So now this is the results. So again, we have used the same data sets, Gita, which is having 50 samples of Parkinson, 50 samples of healthy, and the hyperparameters of SVM, we have used the C and gamma, the different kernel parameters, the linear, polynomial, or RBF. In the random forest classifier, we have used the parameters are the number of estimators. And the PSO, we have used the swarm size of 20, maximum iteration of 200, and the minimum and maximum inertial weight is the 0.4 and 0.9, and the social constant as 2 and 2. Now you see the cost function here, the variation of the cost function over the iteration. You could see here initially the cost function is very high, around 6, then starts reducing until unless we got into the iteration number 15, it is saturated over this point. So this the features, if you look at again how the distribution of the data samples, if you use the reduced features and without reduction of the features, you could see in the right side, bottom right, you see this is the distribution of the samples without the feature selections. So they are quite overlapping. That gives indications that okay, the accuracy could not be better. Now you see in the top left, so this is the distribution of the samples after the selection of the best features, using the best features. So this is the selection of the distribution of the samples. Now you could see here again the samples are very clearly distinguished. The red one is the Parkinson, the green one is the healthy. So now they are separated, they are clustered together. So the variations of the information within the group is now less, and here the variation of the green points are also, they are clustered. The variation is within the class is less. Now between the class is higher because now they have separated. Here is clear-cut separation is there. But when you use all the features to represent a sample, now they are getting overlap. So that is the indication that it reduces the accuracy basically. Now using the only the extracted features, Again, you use the baseline classifier, the support vector machine, and the random forest. I could see in the testing accuracy. Here, all the vowels, you see A, E, I, O, U, the same vowels we have used, and all the vowels, the SVM gives better accuracy compared to the SVM when without using the ANOVA PSO. Okay. The blue one is with ANOVA PSO after a feature selection, and the faded blue is the without ANOVA PSO SVM. Now see, all the dark blue are having high bars, so that means the accuracy is higher compared to all the classes. So it gives an indication that once you use the optimization framework to select the most informative features and then use those features to represent data points, that gives a better accuracy when you use in the support vector machine or random forest classifier. Sorry. So again, if you look at the sensitivity and specificity of each class, again it is giving the better results when you use the feature selection algorithm compared to the without using feature selection algorithm. You see here quite better accuracy in case of SVM as well as in the random forest also. So this gives a summary. In a summary, I can say, so a feature selection algorithm inspired by the social behavior of the swarm sorry, is framed in a ANOVA concept to choose or to select the best informative features. And when you use these best informative features in the PC Gita dataset, that achieves around 98% accuracy, which is quite higher than the classifier without the feature selection. So then the ANOVA PSO feature selection algorithms, this can be used other various applications as optimization approach for different other application in the biomedical application, or where the scenario where you have a less number of data points to be modeled. Okay. So that's it from my side. Thank you for listening to me. If anyone having any questions, I'll be happy to answer.
Dr. Sitantsu, thank you for the nice presentation. Participants, if you have any query, you can write in the chat box.
Let me see the chat box. Let's see here. Yes, this is the chat box.
So if you wish, you can share your screen, Dr. Shah.
Okay, should I stop the screen?
Yes. Stop presentation.
Okay.
So participants, if you have the queries, you put it in the chat box.
I could see.
No. They will be starting.
Okay. So I think now they will be putting the questions.
Yes.
Yeah, I could see one question from Kalpana Joshi. What objective function is used here? Yes, the objective function is the cost function of the objective function is the ANOVA, F score. The F inverse of F score that I have used as a cost function. Okay. So that means I'm trying to minimize the variation of the samples within a class and maximizing the distance of the error between the class. Yeah, that's the objective function. And one other question from Ajay Prajapati. PC Gita dataset is free to use? Yes. but you need to write an email to the the authors. This could be available on a request to the original authors. Yes. I could not see any more question. Okay, another question again from Ajay Prajapati. SBM and RF, which is better? Yeah, so it is it depends on the problem statement that you have. Okay, so sometimes it has been seen that okay, so SBM is better than RF, and sometimes also it has been seen that RF is better than SBM. But it again depends on the data that you have, what type of data you have. Okay, so based on that, your accuracy of the classifier may vary. But yes, you have any categorical data, then random forest will work better than SBM. Yes, but SBM, it has been seen that in binary classification, SBM, it has been seen that SBM is quite good when you have your less number of data points. You have less number of data points, and you have a binary classification classifier at your hand, then SBM is the good choice. Yeah. Can I use deep learning on dataset? Yes, of course. Yes, you can use the deep learning. But it has been seen that many times, basically the deep learning who have a good classification, or the generalized classification, deep learning, you need to have a large amount of data, a variety of data also, which is quite good challenging task in case of biomedical applications. In biomedical field, you don't have much data. That's the real challenge. So that's why people have also used deep learning. Some extent, they have also achieved quite good result, but the reliability is still under mark. So yeah, I prefer if the data set is less, then go for a feature engineering. Okay, extract very good meaningful feature, and also you can use the feature selections. Again, out of the extracted features, select the good one, the best one, informative one. Then use the baseline classifier to classify. Yeah. But if you have a quite good amount of data, then you can use, of course, the deep learning. Yes. I do not see any further question.
Yes.
Dr. Nanda, can you just see from your side?
Yes, there are no further questions. So I thank you for today's presentation as well as for answering all the queries of participants. I further request you to share your slides so that it can be the material can be given
Yes.
to the participants.
Yes, sure. I will send the slides so that you can
Also,
share to the participants. Yeah.
Dr. Siddhansu, I am also very much thankful to you for your continuous contribution in this particular faculty development program, which we started in 2015, and today on 2025, we have almost 10th version of this particular program. So
Thank
thank
you,
you
Dr. Nanda.
for your
So,
support, continuous support for this particular program.
yeah. Thank you for having me continuously in all your series of workshops. Yeah, I'm very much glad to also share my thought and my knowledge to all the participants. Whoever have some kind of benefit from them, yeah, that's my pleasure. Yeah.
Thank you,
So,
Dr. Shah.
thank you so much.
Mhm.